হ্যালো, হ্যালো।
হ্যাঁ স্যার।
হ্যাঁ, পাওয়া যাচ্ছে আওয়াজ?
হ্যাঁ স্যার।
আচ্ছা, ঠিক আছে, আবার চালু করি, আবার চালু, আমার মনে হয় মাঝখানে কানেকশন চলে গেছিল। ওকে, so I will start once more. So what we are talking about a new chapter today. We are starting a new chapter, that is enzymes. So what are enzymes? Enzymes are biological catalysts. They are biological catalysts. So what is the function of a catalyst? A function of a catalyst, it is a catalyst is a substance which increases the velocity or rate of a chemical reaction. The speed of the reaction is increased. Substrate is getting converted into product, but the enzyme itself is not getting converted into something else. It remains as intact as possible. So enzymes may be defined as biological catalysts. They are synthesized by living cells. They are mostly proteins, except RNA like ribozymes, and they are colloidal, thermolabile in character, and specific in their action. Thermolabile means they can be destroyed by heat, except there are the thermophilus aquaticus stack polymerase enzymes. This enzyme is not destroyed by heat. So this enzyme is not destroyed by heat, but others are destroyed by heat, and they are specific. So each enzyme has got a specific function. It acts on a specific substrate or a specific reaction, and it converts that into its product. So this is the classification of an enzyme. Now the definition of an enzyme. And now enzymes can be classified into basically 6 types, and this basically the uh, so you have the term ASE given after an enzyme normally. Say those who break cellulose is known as cellulase. So an ASE is given: fructase, fructose, bisphosphatase, uh, hexokinase. Always an ASE is always given behind it. Okay, and so the basically there are 6 types of enzymes which can be classified. The first is the oxido reductases. Example: alcohol dehydrogenase. Reductase, they catalyze an oxidation and reduction reaction. So an oxidized product is reduced. So AH2 plus B is equal to A plus BH2. So A is oxidized because loss of hydrogen, and B is reduced, that is gain of hydrogen. A becomes from AH2 it becomes oxidized, and B becomes reduced because it gains hydrogen. So this is the function of oxido reductases. Example: alcohol dehydrogenase, the NAD plus oxido reductase. You have other enzymes also. Then you have the enzyme transferases. Example: hexokinase, the first enzyme in the glycolysis. It transfers a group. AX plus B is equal to A plus BX. So this X group is transferred to B. For example, ATP, in ATP, the phosphate is transferred here to uh, glucose to become glucose 6 phosphate. So this is the function of transferases. The next is hydrolyzes. Example of hydrolyzes are lipase. They utilize a molecule of water and they break the water. So AB plus H2O, it becomes AH, and this OH. So AB is broken into AH plus BOH. So hydrolysis. Example: lipase, alkaline phosphatase. These are all hydrolyzes. Then you have enzymes known as lyases. Example, aldolase. You know this aldolase. AB plus XY is equal to AX plus BY. So A and B is broken and this group is attached and this group is attached. So ketose 1-phosphate aldehyde lyase. So you have aldolase, fumarase, histidase. These are enzymes are the lyase. Aldolase, of course, you know, GAPDH and DAP. It breaks fructose 2,6-bisphosphate. Okay, then you have the enzymes isomerases. They convert it into the citrate is converted to converted into isocitrate. This enzyme aconitase. Then you have the triose phosphate isomerase. Glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate in glycolysis. So you have these isomerase enzymes. They convert it into isomers. Then you have the ligases. Basically glutamine synthase. They are condensation reaction, usually dependent on ATP. So A plus B becomes AB. ATP is hydrolyzed into ATP plus PI. So these are the different types of enzymes. Enzymes can be intracellular enzyme or they can be extracellular enzymes. Those who function inside the cell are intracellular enzymes. Those who function outside the cell, they are known as extracellular enzymes. Then there are certain enzyme commission numbers are given. But I would say that forget about enzyme commission numbers over here. Try to concentrate more on the 6 types of classification. Okay, so functional unit of the enzyme is known as holoenzyme, which is often made up of apoenzyme, the protein part, and a coenzyme, the non-protein organic part. So the non-protein organic part is the coenzyme and the functional protein part is the apoenzyme. So holoenzyme, the active whole enzyme, is made up of apoenzyme, the protein part, and the coenzyme, non-protein part. Then another term is used, the prosthetic group. Prosthetic group is used when the non-protein moiety or the non-protein group, it binds covalently and tightly with the apoenzyme. The coenzyme, if you do dialysis, you can separate the coenzyme, but you cannot do that with prosthetic groups that is attached covalently. It is a very strong attachment. You need a chemical reaction to displace it. Then you have monomeric enzymes and oligomeric enzymes. Proteins which act as just one monomer or a single unit, or it can be oligomeric. Several units together make that particular enzyme. So a single monomer can be an enzyme, or it can be a dimer, oligomer, tetramer, pentamer, or pentamer. It can be many things. So enzymes can be this. So you have multi-enzyme complexes also formed, like FAS, fatty acid synthase. They are multi-enzyme complexes. Now, the next thing which you study is effect of enzyme concentration on enzyme velocity. So if for a reaction A to B, how fast A is becoming B, so your enzyme velocity, it is measured as amount of product formed per unit time, not the amount of distance covered in unit time, amount of product formed from substrate in unit time, or the amount of substrate which disappears to form product per unit time. So you do this particular thing. So you have enzyme velocity, and enzyme concentration. So gradually, as you increase the enzyme concentration, you can see that the velocity of the reaction, enzyme reaction, it increases. So what are the factors which affect enzyme activity? We will now discuss about the factors which affect enzyme activity. The factors which affect enzyme activity are the first, the concentration of enzymes. As you had seen that the concentration, the more you increase the concentration of enzyme, the faster the reaction takes place. The faster the reaction takes place, the more you add enzymes. Next is concentration of substrate. Now if we look at the concentration of substrate, this is substrate concentration and this is velocity. So as you increase, keep on increasing the substrate concentration, if you keep on increasing the substrate concentration, the velocity keeps on increasing till it reaches a maximum velocity, after which it the rate of the velocity of the reaction doesn't increase. So this is known as Vmax, where the maximum velocity is Vmax, and half of Vmax is this half of this value. And you have another term known as Km. This Km essentially is the substrate concentration at which half of Vmax is reached. So you bring it to this line and bring it, plot it down. This is the substrate concentration at which half of Vmax is reached. So this is the essentially this is how the effect of substrate concentration takes place. Now there is a proof which you have to practice over here. I will send you my note where you can find I've calculated over there. I will not waste time on that because mostly it doesn't come in that exam. So I will rather come what you have to understand what is Km, what is the what does low Km mean and what does high Km mean. So if Km value is low, say if the Km value is low, then the reaction rate is like this, is more steeper. Low Km means the enzyme has got higher affinity for the substrate. So the curve becomes like this. That means higher affinity for the substrate means it will bind to it much more better. And say if the Km is higher over here, that means bring it over here. So this is going to take the velocity is much more, it is going to take a longer time. You need more substrate over here. So higher Km means enzyme affinity for the substrate or the rate is much less, and lower Km means it's very high. So this is the philosophy of Km. Now Km is known as the Mikhailis maintenance equation. Now let us have a look at the Mikhailis maintenance equation. Mikhailis maintenance constant. It is also known as the Briggs-Haldane constant. After Briggs as well as JBS Haldane. So or first order reaction, this is a first order reaction. So order of when the velocity of the reaction is almost proportional to the substrate concentration, That is, the more substrate you give, the more the enzyme velocity is respond. That is the first order reaction. That is known as the first order reaction. And when the concentration of substrate is much greater than Km, the rate of the reaction is independent of substrate concentration, and it is known as zero order reaction. When it is not, the rate of the reaction is not dependent on the substrate concentration. Yeah. So let us look at this reaction E plus S. The reaction is ES, and you get a product E plus P. This is a transition state. ES is a transition state. Now E plus enzyme plus substrate, it has got a rate constant, and that rate constant is known as K1. Even ES, there is the back reaction which takes place with the rate constant K2, and what you get is E plus S there. And you have the enzyme substrate transient complex gets broken into enzyme plus product, and that is K3. That it has got a rate constant K3. In this equation they are ignoring this reverse reaction assumption. So K1, K2 and K3, they represent the velocity constant for the reaction, and Km is K2 plus K3 divided by K1. This is the Briggs-Haldane constant, and Mikhailis and Maintenance empirically got this formula. This is known as the Mikhailis-Maintenance equation. V max into concentration of substrate divided by Km plus V, the velocity of a reaction. V is the measured velocity, V max is the maximum velocity which we have seen, S is the substrate concentration, and Km is the Mikhailis Maintenance equation. So remember these formulas. Now when you look at this particular curve, this is a parabolic curve, hyperbola curve, sorry, not a para. So in such a hyperbola curve, you always have, in the maximum, it is always reaching the maximum, but it is still 1 point less. It is tending towards reaching the maximum speed, but still it has not reached the maximum speed. So in such a case, you can't get, in this particular curve, you cannot get the value of this hyperbolic curve value, accurate Vmax value. If you don't get an accurate Vmax value, it is almost more or less it's the same, but it is not accurate, because it is almost tending towards the maximum, but it has not reached the maximum. So it is always reaching, it's always there. Whatever is the final value, it is just a bit less than that. So in this case, practically when you measure this on a graph, when you are doing this particular expression, you have different substrate concentration, a fixed enzyme concentration, and then you have got a fixed velocity. You are doing this particular reaction. It is very difficult to do this particular reaction at that point of time. So what you do, there is a Lineweaver-Burke have found out a double reciprocal curve. So what he did was, he divided this by V, and he divided this by all by 1. So this this becomes 1 by V, and here it becomes 1 by substrate concentration. He does a double reciprocal plot. So once you use a double reciprocal plot, 1 by V and 1 by S, it becomes like this. You get, instead of the hyperbolic curve, you get a straight line, and this point becomes the Vmax, and this is minus 1 by Km, and the slope is Km by Vmax. So you get these values. So when you are doing practically you are doing the experiment, this Lineweaver-Burke curve, it will help a lot in your work. So now let us see the effect of temperature on enzyme activity. So effect of temperature, more the temperature increases, the more enzyme activity, till it reaches a maximum known as optimum, after which if you heat, the enzyme denatures and the activity decreases down. It's a bell shaped curve. The same thing happens with that of increasing pH. Increasing pH, the enzyme activity increases, and then after certain time, more increasing pH, it means that it will start to fall. Other way around, if you look, decrease it, it comes to the maximum optimum, and then it will fall. So enzymes in our digestive system, for example, enzymes in our stomach and enzymes in the intestine. You know, stomach is acidic and intestine is alkali. So enzymes in the stomach, they have an optima which is more on the acidic range. So maximum activity will be found when your stomach HCl is secreted. You will get the maximum optimal. But intestinal enzymes, they have got an optima at an alkaline pH. So the enzyme optima is different. So your stomach enzymes will not work in your intestine, and your intestinal enzymes will not work in the stomach. So digestion in stomach and intestine are fantastically kept separate by just utilizing this pH optima. Now, effect of product concentration. So more the product, the speed of the reaction will decrease, because you don't need, this is known as product inhibition. More product is formed, then it will inhibit the enzyme to make more product, because there is sufficient product is formed. For example, lot of citrate, Krebs cycle is inhibited because there is lot of citrate. Why will more pyruvate be converted to citrate? Because you have sufficient. Then you have effect of activators. So an enzyme has got an active site in which the substrate fits, and it needs certain metallic cations like magnesium, manganese, zinc, bivalent predominantly for their activity. Light and radiation, they sometimes they damage enzymes also. Then you have the what is the active site of the enzyme. This is the active site where the real enzyme activity or the real enzyme fun takes place. That is the active site. So the active site or active center of an enzyme represents the small region at which the substrate binds and participates in the catalysis. So that is the active site. So today I will stop my class over here. Next day we will study about enzyme inhibition. So if you have any questions, please do not hesitate to ask me. Do you people have got any questions? So there are 19 students over here. Orpita, Hina, Jayanti, Kiran, then Monidipto, Megha, Papia, Porichita, Shambhavi, Sanchari, Saurabh Garai, Shreya, Siddharth Bhomik, Moinuddin, Sneha Chakraborty, Suparna Layak, Sushmita Pal, and Zakia Parveen. So these are the people who are in the class. Okay, so there are 19, that means 18 students are present today. So any questions, please do ask. Do you people have any questions?
স্যার
হ্যাঁ
হ্যাঁ বলছিলাম যে আমরা এনজাইম নমেনক্লেচারটা পড়বো না, যে এটা মানে একটা ইন্টারন্যাশনাল কোড
না
অফ
না
নমেনক্লেচার ফর
1.1
এনজাইম
ওটা
আছে।
দরকার নেই, আর পড়ার দরকার নেই।
আচ্ছা আচ্ছা
ওটা আমি ওই 6 বছর আগে প্রশ্নটা দেখেছি, তারপরে আর দেখিনি। এনজাইম
আচ্ছা
নমেনক্লেচার পড়তে গেলে আমি তোমাদের একটা দিয়ে দেবো, ওটা মুখস্থ করে রেখে দেবে। ওটা এখন পড়তে গেলে ও অনেক টাইম লাগবে তোমাদের। ওটা আসেও না প্রশ্ন, পড়ে কোনো মানেও হয় না। 1.2.c এবারে ওগুলো, হ্যাঁ ওগুলো পড়ার কোনো মানে হয় না ইন মাই ওপিনিয়ন। এখন অন্য স্যারেরা বলবে
আচ্ছা
যে হ্যাঁ পড়তে হবে, কিন্তু আমি
না না ঠিক আছে।
আমি যেহেতু লাস্ট মানে এই প্রশ্নটা আসবেও না, তোমাদের আসবে না এটুকু আমি গ্যারান্টি দিতে পারি। কারণটা
আচ্ছা
আমি আর
আচ্ছা
বলবো না, কিন্তু তোমার লাস্ট 4-5 বছর ধরে আসেনি। তবে what are the function of oxido reductase, what is the function of lyase, what is the function of transferase, এইগুলো আসবে।
হ্যাঁ, ওগুলো যেহেতু আমাদের
ওই
মেটাবলিক
ছটা
পাথওয়েজ গুলোতে
হ্যাঁ হ্যাঁ ওইগুলো আসবে, ওই
ইনক্লুড আছে
ছটার মধ্যে
তাই জন্য।
ওইটা আসতে পারে। কিন্তু ওই এই এনজাইমটা কি, এনজাইম 2+3 2.1.2.1 এই জিনিসগুলো কি, এগুলো মোস্ট প্রবাবলি আসবে না, আসলেও বাদ দিয়ে দেবে, ওটা করতে হবে না।
ঠিক আছে।
কেন ওটা অনেক মানে তোমাদের ওর পেছনে যতটুকু আসার চান্স আছে তোমাদের খাটনিটা বেশি হবে, কস্ট বেনিফিটটা দেখতে হবে তোমার। তুমি যতটা এফার্ট দিচ্ছ তার রেজাল্টটা তুমি কি পাবে। যেরকম ফর এক্সাম্পল তোমাদের প্রোটোজোয়ার ক্লাসিফিকেশন, ঠিক আছে,
হুম হুম
তোমাদের তো আপ
হুম।
টু ক্লাস আছে, এবারে আপ টু
হ্যাঁ
ক্লাস প্রোটোজোয়ার ক্লাসিফিকেশন তোমাদের যতটা অবধি এতগুলো ক্লাসিফিকেশন, তো তোমাদের এতগুলো অর্ডার, হ্যাঁ মনে রাখা, তোমাদের ফিশের ক্লাসিফিকেশন 44খানা তোমার জাস্ট,
হ্যাঁ, অনেকটা বড় আছে
হ্যাঁ
ফিশের
ফিশের
ক্লাসিফিকেশনটা
ক্লাসিফিকেশন
বুঝলাম।
এইগুলো, এটার পেছনে তুমি যতটা এফার্ট দেবে তার রিটার্নটা হয়তো দেখবে 1 নম্বর কি 2 নম্বর আসছে, তার 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 রিটার্নটা তো ভালো পাচ্ছ না। সুতরাং মানে মানে বুদ্ধিমানের কাজটা হচ্ছে যে এটাকে বাদ দাও, রাদার অন্য জিনিস যেগুলো আরো দরকারি, যেগুলো করা খুব দরকার, যেরকম তোমার এনজাইমের অ্যাক্টিভ সাইট কি, হোয়াট আর দ্য এফেক্ট অফ টেম্পারেচার, পিএইচ, এটা করা,
হুম হুম হুম।
মাইকেলিস মেনটেন্স ইকুয়েশন, হোয়াট ই ভিম্যাক্স, কেএম, এইগুলো ইম্পর্টেন্ট, এনজাইম ইনহিবিশনটা ইম্পর্টেন্ট, অ্যালোস্টেরিক রেগুলেশন কিভাবে হয় এগুলো ইম্পর্টেন্ট, এইগুলো পড়ে নেওয়াটা তখন আমার মতে ওইগুলো আরো ইম্পর্টেন্ট হয় তখন,
ঠিক আছে স্যার।
রাদার ওই 1 নম্বরের পেছনে মানে সে ট্রিমেন্ডাস এফার্ট দিয়ে। তবে অ্যামাইনো অ্যাসিডের যেরকম তোমার 20টা অ্যামাইনো অ্যাসিডের থেকে ওটা তো ইজিলি মনে থাকা যায়। হ্যাঁ
হ্যাঁ,
ওই,
ওটা তো যেহেতু আমরা এইচএসেও
হ্যাঁ
পড়েছিলাম তো
ওই তিন
অসুবিধা
চারটে
হবে না।
বাদ দিয়ে তুমি বাকি অ্যামাইনো অ্যাসিডগুলো মনে রাখা যায়, ওই অ্যারোম্যাটিক
হুম।
ওইগুলো একবার করে লিখলেই হয়ে যায়, ওটাতে বেশি এফার্ট লাগে না।
হ্যাঁ হ্যাঁ।
তাই জন্য বলবো ওইগুলো করে রাখতে। হ্যাঁ বলো, বলো স্নেহা, বলো কি বলছিলে, হ্যালো বলো।
কিছু বলছিলাম না স্যার, ও ভুল করে স্যার আনমিউট হয়ে গেছে।
আচ্ছা ঠিক আছে, আর কারুর কোনো কোয়েশ্চেন আছে? না থাকলে তাহলে আজকে আমি শেষ করছি, বাই বাই, ভালো থেকো। ওকে।
হ্যাঁ স্যার।
হুম হুম।